back to Anatomiqa. In this session, we will be discussing the details of the development of heart. The learning objectives of the session are: describe the cardiogenic area and its relation to the pericardial cavity, describe the formation of the heart tube, its parts and looping, describe the formation of the interatrial septum, describe the formation of the right and left atria, describe the changes in the sinus venosus, describe the formation of the atrioventricular valves, describe the formation of septa in truncus, conus and ventricle, describe the formation of the two ventricles, describe the formation of the semilunar valves, describe the formation of the conducting system of the heart, describe the formation of transverse and oblique sinuses of the pericardium, describe the derivatives of the vitelline, umbilical and cardinal veins. Give examples of congenital malformations with special reference to atrial septal defects, ventricular septal defects, Fallett's tetralogy, and arterial and venous systems. Now, to begin with, establishment and patterning of primary heart field. In the middle of the third week, the embryo is no longer able to satisfy the nutritional requirements by diffusion. So, to satisfy the nutritional requirements, the vascular system starts developing. The progenitor heart cells lie in the epiblast immediately adjacent to the cranial end of the primitive streak. From there, they migrate through the streak and into the splanchnic layer of lateral plate mesoderm, where they form a horseshoe-shaped cluster of cells called the primary heart field, cranial to the procaudal plate. As the progenitor heart cells migrate and form the primary heart field during 16 to 18 days, the cells are specified on both sides from lateral to medial to become atria, left ventricle and most of the right ventricle. The remainder of the heart, including part of the right ventricle and outflow tract, that is the conus cordis and the truncus arteriosus, is derived from the secondary heart field. The cells of the secondary heart field appears slightly later during 20 to 21 days than those in the primary heart field. They reside in splanchnic mesoderm, ventral to the posterior pharynx, and is responsible for lengthening the outflow tract. Cells in the secondary heart field also exhibit laterality like that of the primary heart field. The cells on the right side contribute to the left of the outflow tract region, and the cells on the left side contribute to the right of the outflow tract. This explains the spiraling nature of the pulmonary artery and aorta and ensures that the aorta exits from the left ventricle and the pulmonary artery from the right ventricle. Coming to the formation and position of the heart tube, initially the central portion of the cardiogenic area is cranial to the precaudal plate or oropharyngeal membrane or buccopharyngeal membrane and the neural plate. With closure of the neural tube and formation of the brain vesicles, however, the central nervous system grows so rapidly towards cranial end that it extends over the central cardiogenic region and the future pericardial cavity. As a result of outgrowth of the neural tube and cephalic folding of the embryo, the precaudal plate or oropharyngeal membrane is pulled forward while the cardiogenic area, which develops into heart and pericardial cavity, move first to cervical region and finally to the thoracic region. Once the epiblast cells establishes the primary heart field, the underlying pharyngeal endoderm includes the cells in the primary heart field. Now, the cells in the primary heart field forms cardiac myoblasts and blood islands that will further form blood cells and vessels by the process of vasculogenesis. Let us see how the primitive heart tube arises from vasculogenesis. At the cephalic end of the embryonic disc in the primary heart field, cephalic to the site of the developing mouth and the nervous system, clusters of cells arise in the mesenchyme. These cluster of cells are called as blood islands and the cells are called as angioblasts. The angioblasts now differentiate into two groups of cells, outer cells which forms the endothelial cells and the inner cells which form hemocytoblasts, giving rise to blood cells. Gradually, the endothelial cells fuse with each other called endothelial channels between the adjacent blood islands. This forms a continuous channel in the form of horseshoe-shaped structure, giving rise to the primitive heart tube or the endocardial heart tube. The endothelial lined tube is surrounded by myoblasts. In addition to the cardiogenic region, other blood islands appear bilaterally, parallel and close to the midline of the embryonic shield. These islands form a pair of longitudinal vessels called the dorsal aortae. Now these are the two heart tubes on the right side and the left side of the embryonic disc developing in the splanchnopleuric mesoderm. As the embryo grows and bends laterally, the two cardiac tubes merge to form single primitive heart tube. Thus, the heart becomes a continuous expanded tube consisting of an inner endothelial lining and an outer myocardial layer. Now, it receives venous drainage at its caudal pole and begins to pump blood out of the first aortic arch into the dorsal aorta at its cranial pole. Now let us see the relationship between the growing heart tube and the pericardial cavity. The pericardial cavity is developing from the intraembryonic coelom. In the beginning of the development, the cardiogenic area is at the cranial end of the embryonic disc in the cardiogenic area. The developing heart is ventral to the developing pericardial cavity. With the cranial folding of the embryonic disc, the position and relationship of the structures changes. The cardiogenic area migrates ventrally and caudally towards the thoracic region. The developing heart now becomes positioned dorsal to the developing pericardial cavity. As the heart tube starts to grow bigger, it bulges into the pericardial cavity. As the developing heart tube bulges more and more into the pericardial cavity, initially the tube remains attached to the dorsal pericardial cavity by a fold of mesodermal tissue called the dorsal mesocardium. No ventral mesocardium is ever formed. Simultaneously, it becomes surrounded by a thick layer of mesenchyme in the splanchnopleuric mesoderm, which differentiates into the myocardium and the visceral layer of the serous pericardium. The somatopleuric mesoderm develops into the parietal layer of serous pericardium and fibrous layer of pericardium. The cavity in between the two serous layers of pericardium forms the pericardial cavity. The arterial end of the primitive heart is continuous beyond the pericardium with a large vessel, the aortic sac, and the venous end, that is venous sinuses, is also outside the pericardium. With further development, the dorsal mesocardium starts disappearing and completely disappears, creating the transverse pericardial sinus, which connects both sides of the pericardial cavity. The heart is now suspended in the pericardial cavity and is attached to the pericardium only at its ends. At the cranial end, it is attached to the aortic sac, and at the caudal end to the sinus venosus. In the diagram on the left, which is the cross section of the embryo at the level of formation of heart, shows the dorsal mesocardium, which later disappears and becomes the transverse pericardial sinus. During these events, the myocardium, developed from splanchnic mesoderm, thickens and secretes a thick layer of extracellular matrix rich in hyaluronic acid called cardiac jelly. This cardiac jelly separates the myocardium from the endothelium of the primitive heart tube. In addition, mesothelial cells on the surface of the septum transversum form the proepicardium near the sinus venosus at its caudal end and migrate over the heart to form most of the epicardium. The remainder of the epicardium is derived from the mesothelial cells originating in the outflow tract. Thus, the heart tube consists of 3 layers. All the 3 layers develop from the splanchnopluric layer of lateral plate intraembryonic mesoderm. The innermost layer is the endocardium, forming the internal endothelial lining of the heart. The myocardium forming the muscular wall, and the epicardium forming the visceral layer of serous pericardium covering the outside of the tube. This outer layer is responsible for formation of the coronary arteries, including their endothelial lining and smooth muscle. Let us now see what is the fate of the primitive heart tube, how it happens. The heart tube undergoes differential expansion so that several dilatations separated by grooves are formed. While the local expansions become visible throughout the length of the tube, simultaneously the heart undergoes looping. From the arterial to the venous end, these dilatations are called the bulbus cordis, the primitive ventricle, the primitive atrium, and the right and left horns of the sinus venosus. The bulbus cordis and the ventricular parts of the tube now elongate more rapidly than the remainder of the tube, and since the arterial and venous ends are fixed by the pericardium, the tube begins to bend. As the outflow tract, that is bulbus cordis, lengthens, the cardiac tube begins to bend on day 23. The cephalic portion with bulbus cordis of the tube bends ventrally, caudally and to the right, and the atrial or the caudal portion shifts dorsally, cranially and to the left. This bending, which may be due to cell shape changes, creates the cardiac loop. The bend soon becomes U-shaped and then forms a compound S shape, with the atrium lying posterior to the ventricle. Thus, the venous and arterial ends are brought close together as they are in the adult. The looping of the heart is completed by day 28. As these changes are taking place, a gradual migration of the heart tube occurs from the neck region to the thoracic region. The passage between the atrium and the sinus venosus forms the sinoatrial orifice. The junction between the atrium and the ventricle narrows to form the atrioventricular canal. The atrial portion, initially a paired structure present outside the pericardial cavity, forms a common atrium and is incorporated into the pericardial cavity. The bulbous cordis divides into three parts. The proximal portion bulges into large cavity and is called the proximal 1/3 of bulbous cordis. This portion does not have a separate name and will form the trabeculated part of the right ventricle. The mid portion, the conus cordis, will form the outflow tracts of both ventricles. The distal part of the bulbous cordis, called the truncus arteriosus, will form the roots and the proximal portion of the aorta and pulmonary artery. The junction between the ventricle and the bulbous cordis, externally indicated by the bulboventricular sulcus, remains narrow. It is called the primary interventricular foramen or the bulboventricular canal. Thus, the cardiac tube is organized by regions along its craniocaudal axis as conotruncus, right ventricle, left ventricle, and atrial region respectively. To get an orientation of how the looping of heart takes place, watch this animation. So, after looping of the heart, the positions of the chambers are as seen in these lateral and frontal views. The arterial end, represented by bulbous cordis, bends ventrally caudally and to the right. So, the right ventricular chamber is present in front and to the right of all other chambers of the heart. The primitive ventricle remains in the middle, so the left ventricle is slightly behind the right ventricle and to the left of it. The venous end, represented by the primitive atrium and sinus venosus, bends dorsally, cranially and to the left, so the right and left atria are observed on the dorsal and cranial aspect of the ventricles. Sinus venosus is observed in the lateral view, not in the anterior view, as this is present on the most dorsal aspect of the heart tube. Circulation through primordial heart. By this time, the heart is functionally ready for the blood circulation. Blood enters the sinus venosus from the embryo through the common cardinal veins, developing placenta through the umbilical veins, umbilical vesicle through the vitelline veins. Blood from the sinus venosus enters the primordial atrium. The flow from the atrium is controlled by the sinoatrial valves. The blood then passes through the atrioventricular canal into the primordial ventricle. When the ventricle contracts, blood is pumped through the bulbous cordis and truncus arteriosus into the aortic sac, from which it is distributed to the pharyngeal arch arteries. The blood then passes into the dorsal aortae for distribution to the embryo, umbilical vesicle and placenta. Looking into the development of each part of the heart tube, first let us see the development of sinus venosus and its derivatives. During the 4th week of development, the sinus venosus is connected to 3 veins in the right side and left side. They are vitelline veins, which return blood which is poorly oxygenated from the umbilical vesicle or yolk sac. Umbilical veins, which carry well oxygenated blood from the chorionic sac. The right umbilical vein degenerates and only the left umbilical vein persists. Common cardinal veins return poorly oxygenated blood from the body of the embryo to the heart. It has 2 tributaries. Anterior cardinal vein, which drains blood from the cranial end of the embryo, and posterior cardinal vein, which drain blood from the caudal end of embryo, and both join to form the common cardinal vein and drains into the sinus venosus. In the middle of the 4th week, the sinus venosus receives venous blood from the right horn and the left horn of sinus venosus. We have already discussed about the veins draining into the right and left horn of sinus venosus. To repeat, each horn receives blood from 3 important veins: the vitelline or the omphallomesenteric vein, the umbilical veins, and the common cardinal veins. At first, communication between the sinus venosus and the primitive atrium is wide, communicating between the atrium and the 2 horns of sinus venosus, and this is called the sinoatrial orifice. As the development proceeds in the 5th week, the left sinus horn rapidly loses its importance with the obliteration of the right umbilical vein, the left vitelline vein, and in the 10th week, the left cardinal system obliterates. Due to these obliterations, the blood entering the left horn of sinus venosus is decreased and there is left to right shunts of blood. The size of the left horn of sinus venosus is reduced and it becomes a tributary to right horn of sinus venosus. The sinoatrial orifice is shifted to the right side and is restricted between the atrium and the right horn of sinus venosus. As a result of left to right shunts of blood, the right sinus horn and veins enlarge greatly. Its entrance, the sinoatrial orifice, is flanked on each side by a valvular fold, the right and left venous valves. The right horn, which now forms only the communication between the original sinus venosus and the atrium, is incorporated into the right atrium to form the smooth-walled part of the right atrium. The reduced part of the left horn of sinus venosus forms the oblique vein of the left atrium and the coronary sinus. Dorsocranially, the right and left venous valves fuse to form a ridge known as the septum spurium. We will discuss the fate of the valves later after discussing the formation of interatrial septum. Fate of the veins draining into the sinus venosus in the thoracic part is discussed now. The rest will be discussed respectively during the development of head and neck and the abdomen. The left vitelline vein mostly obliterates and the remaining part gives rise to the portal vein. The right vitelline vein develops into the hepatic veins and the caudal portion of the inferior vena cava. The cranial part of the left umbilical vein between the liver and the sinus venosus degenerate. The persistent caudal part of the left umbilical vein becomes the umbilical vein connecting the fetus and the placenta, carrying well-oxygenated blood from the placenta to the fetus. The right umbilical vein completely degenerates. The 2 anterior cardinal veins anastomose with each other. Left cardinal vein above the anastomosis and the anastomotic channel give rise to the left brachiocephalic vein. The rest degenerates. The right anterior cardinal vein above the anastomosis gives rise to the right brachiocephalic vein and below the anastomosis develops into part of superior vena cava. The left posterior cardinal vein forms part of inferior vena cava. The right posterior cardinal vein gives rise to root or arch of azygous vein opening into superior vena cava. The left common cardinal vein obliterates. Right common cardinal vein develops into part of superior vena cava. We have completed the development of sinus venosus and its tributaries. Now let us discuss the development of the right and left atria from the primitive atrium, which begins approximately in the middle of the 4th week and is completed by the end of the 8th week. The two atria develop from different sources. The common sources contributing in the formation of the right and left atria are formation of interatrial septum, primitive atrium, and atrioventricular canals. The sources which contribute for the development only to the right atrium is right horn of sinus venosus, and only to the left atrium is pulmonary veins. Formation of the cardiac septum. I just want to insist that the development of different parts of the heart are developing simultaneously and not part by part. Please understand this before we proceed. Just for our understanding, we are going part by part. So by the end of the 4th week, in the atrioventricular canal, endocardial cushions form on the dorsal and ventral walls. These cushions approach each other and fuse to form septum intermedium, dividing the AV canal into right and left atrioventricular canals. These canals separate the primitive atrium from the ventricle, and these cushions function as atrioventricular valves. We will discuss this in detail later. Now coming to the development of septum formation in the common atrium, at the end of the 4th week, a sickle-shaped crest grows from the roof of the common atrium into the lumen. This crest is the first portion of the septum primum. The two limbs of this septum extend toward the endocardial cushions in the atrioventricular canal. The opening between the lower rim of the septum primum and the endocardial cushions is the ostium primum or the foramen primum. With further development, the septum primum grows further caudally. Extensions of the superior and inferior endocardial cushions grow along the edge of the septum primum. Closing the foramen primum. Before closure is complete, however, cell death or apoptosis produces perforations in the upper portion of the septum primum. Coalescence or joining of these perforations forms the ostium secundum or foramen secundum, ensuring free blood flow from the right to the left primitive atrium. In this slide, you can see that the size of the foramen primum is reduced and the small spaces above the septum primum are fused to form a large space forming the foramen secundum. At this stage, both foramen primum and foramen secundum are present. The endocardial cushions are fused to form the septum intermedium. When the lumen of the right atrium expands as a result of incorporation of the sinus on, a new crescent-shaped fold appears. This new fold, called the septum secundum, never forms a complete partition in the atrial cavity. Gradually, the free edge of the septum secundum begins to overlap the foramen secundum. The anterior limb of the septum secundum has extended onto the septum intermedium. The septum secundum now has overlapped the foramen secundum and the upper margin of the septum primum. Now a gap appears between the septum primum and septum secundum. This gap is called the foramen ovale. The upper part of the septum primum gradually disappears and the remaining part, which is on the lower aspect, becomes the valve of the oval foramen. Now, the upper part of the septum primum has disappeared. The remaining lower part is acting as the valve of the foramen ovale. Before birth, the foramen ovale allows oxygenated blood that has entered the right atrium from the inferior vena cava to pass into the left atrium. The lower part of the septum primum, acting as a flap-like valve, opens up and allows blood to pass into the left atrium but does not allow blood to enter into the right atrium. It prevents blood from moving from the left atrium into the right atrium. After birth, in the normal development, when lung circulation begins and pressure in the left atrium increases, the valve of the oval foramen is pressed against the septum secundum and fuses with it, obliterating the foramen ovale and separating the right and left atrium. The two atria thus are separated from each other. The lower edge of the septum secundum, seen in the right atrium, becomes the annulus ovalis or the limbus fossa ovalis, and the depression below this is called the fossa ovalis, which is the remnant of septum primum. The right and left auricular appendages later develop as small diverticula from the right and left atria respectively. Now, coming to the clinical aspect of the foramen ovale, probe patency of foramen ovale belongs to atrial septal defects. In about 25% of cases, fusion of septum primum and septum secundum is incomplete, and a narrow oblique cleft foramen ovale remains between the two atria. This condition is called probe patency of the foramen ovale. In this condition, there is no intracardiac shunting of blood. This probe patency is usually of no clinical importance. Coming to the atrial septal defects, the probe patency of the foramen ovale is discussed previously, which is caused by incomplete anatomic fusion of septum primum and septum secundum. Premature closure of the foramen ovale is closure of the foramen ovale during prenatal life. It results in hypertrophy of the right side of the heart and underdevelopment of the left side of the heart. Foramen secundum defect is caused by excessive resorption of septum primum, septum secundum, or both. This results in a condition in which there is an opening between the right and left atria. Some defects can be tolerated for a long time, with clinical symptoms manifesting as late as age 30 years. Common atrium, also called cor trilocular biatriatum, is caused by the complete failure of septum primum and septum secundum to develop. This results in a condition in which there is formation of only one atrium. For more details of the clinical aspects, check the video on the congenital heart defects. Till now, we have seen the development of the interatrial septum, dividing the primitive atrium into right and left parts. Coming to the other sources contributing to the development of right atrium, that is, incorporation of the right horn of sinus venosus. The left horn of sinus venosus is reduced in size and has migrated its opening from right atrium to the right horn of sinus venosus, forming a tributary to it. The left horn of sinus venosus later develops into coronary sinus. The right vitelline vein develops into terminal part of inferior vena cava, and the right common cardinal vein develops into intrapericardial part of superior vena cava. In the meantime, as they are developing, right horn of sinus venosus is absorbed into the right atrium as the cavity enlarges to form the smooth part of the right atrium. With the incorporation or absorption of right horn of sinus venosus into the wall of right atrium, the newly formed coronary sinus, inferior vena cava and superior vena cava now directly open into the right atrium. The left venous valve of the sinoatrial orifice merges with the interatrial septum, and the right venous valve of the sinoatrial orifice forms the crista terminalis. The details of the sinoatrial orifice will be seen in the next slide. Now, we are focusing on another source contributing to the development of right atrium, that is the venous valves of the sinoatrial orifice. The two valves of the sinoatrial orifice, the right venous valve and the left venous valve, dorsocaudally fuse with each other to form the septum spurium. Initially, the valves are large, but when the right horn of sinus venosus is incorporated into the wall of the right atrium, the left venous valve and the septum spurium merge with the septum secundum of the developing atrial septum. The superior portion of the right venous valve forms the crista terminalis, which divides the anterior rough part and the posterior smooth part of the right atrium. The inferior portion develops into two parts, the valve of the inferior vena cava and the valve of the coronary sinus. Hence, on the whole, the right atrium is developed from the following sources: posterior smooth part from the absorption of right horn of sinus venosus, anterior rough part from the primitive atrium, right half of the atrioventricular canal, right venous valve will give rise to crista terminalis, valve of inferior vena cava and valve of coronary sinus. Coming to further differentiation of left atrium, the primitive left atrium is likewise expanding concurrently as the primitive right atrium was enlarging by incorporation of the right horn of sinus venosus. Initially, a single embryonic pulmonary vein develops as an outgrowth of the posterior left atrial wall, just to the left of the septum primum. There are two views related to the development of the pulmonary veins. According to the first view, the pulmonary vein develops as a sprout and gains connection with veins of the developing lung buds. The second view is that the pulmonary vein sprouts and bifurcates to form four veins, which then grows towards the lung buds. During further development, as seen in the diagrams, the pulmonary veins and its branches are incorporated into the left atrium. Although initially one vein enters the left atrium, ultimately four pulmonary veins enter as the branches are incorporated into the atrial wall, resulting in the direct opening of all four pulmonary veins into the wall of the left atrium. As the left atrial wall expands, the smooth tissue of the pulmonary veins is incorporated into the wall, forming the smooth part of the left atrium and displaces the trabeculated tissue anteriorly and laterally, which will then form the adult auricles. So, in the fully developed heart, the original primitive left atrium is represented by little more than the trabeculated atrial appendage, while the smooth walled part originates from the pulmonary veins. This diagram shows the section of the two atria at the level of incorporation of the four pulmonary veins into the left atrium. Observe how one pulmonary vein opening into the wall gradually with development leads to direct opening of four pulmonary veins due to the absorption of the walls of pulmonary veins, leading to smooth walled left atrium. Finally, the left atrium develops from the following sources. Posterior smooth part from the incorporation of four pulmonary veins. Anterior rough part develops from the primitive atrium and left half of atrioventricular canal. Now that we have completed the development of sinus venosus and its tributaries and the development of the right and left atria, which is the venous end of the heart, we will now focus on the development of the arterial end of the heart. It includes the development of atrioventricular orifice or canal, primitive ventricle, bulboventricular canal, bulbus cordis, dividing into three parts: proximal 1/3 of bulbus cordis, conus cordis, and truncus arteriosus. The atrioventricular canal divides into tricuspid and bicuspid orifices with respective valves. Primitive ventricle develops into left ventricle. Bulboventricular canal is closed by the development of interventricular septum. Proximal 1/3 of bulbus cordis gives rise to right ventricle. The conus cordis and truncus arteriosus give rise to pulmonary trunk and aorta. Let us start with the septum formation in the atrioventricular canal. Initially, the atrioventricular canal gives access only to the primitive atrium, developing into right and left atria, and the primitive ventricle, which develops into only left ventricle. But the atrioventricular canal is separated from the bulbus cordis, which is going to develop into the right ventricle by the bulboventricular or conoventricular flange. It is nothing but a ridge separating the two parts. Near the end of the 5th week, the posterior extremity of the ridge terminates almost midway along the base of the superior endocardial cushion and becomes less prominent than before. The atrioventricular canal enlarges to the right, and the blood passing through the atrioventricular orifice now has direct access to the primitive left as well as the primitive right ventricle. Let us start with the septum formation in the atrioventricular canal. At the end of the 4th week, in the common atrioventricular canal, two mesenchymal cushions, the anterior and the posterior atrioventricular endocardial cushions, appear at the anterior and posterior borders of the atrioventricular canal. In addition to the anterior and posterior endocardial cushions, the two lateral atrioventricular or endocardial cushions appear on the right and the left borders of the canal. In the meantime, the anterior and posterior cushions project further into the lumen and fuse, resulting in the formation of complete septum called septum intermedium, dividing the canal into right atrioventricular and left atrioventricular canals by the end of 5th week. Now the blood from the right atrium passes through the right atrioventricular orifice into right ventricle. The blood from the left atrium passes through the left atrioventricular orifice into the left ventricle. Now that we have seen the formation of atrioventricular canals, we need to know how the atrioventricular valves are formed. After the septum intermedium is formed, each atrioventricular orifice near its margin is surrounded by local proliferations of mesenchymal tissue covered by endocardium. When the blood starts flowing into the ventricle, it starts eroding the muscle tissue and thins the tissue on the ventricular surface of these proliferations. Thinning of the tissue on the ventricular valves leads to formation of atrioventricular valves. As the mesenchymal tissue in the valves are replaced by fibrous tissue, the valves now consist of connective tissue covered by endocardium. With the erosion of the muscular tissue in the walls of the ventricle by the blood flow, at intervals the valves remain attached to the ventricular wall by muscular strands or cords. Later, the mesenchymal tissue in the muscular strands is replaced by the fibrous tissue and becomes differentiated into chordae tendineae and papillary muscles. Finally, the valves are attached through the chordae tendineae to the papillary muscles. Hence, two valve leaflets constituting the bicuspid or mitral valve form in the left atrioventricular canal and three constituting the tricuspid valve form on the right side. Coming to the clinical considerations, the atrioventricular septal defects are persistent common atrioventricular canal. This is caused by failure of fusion of the dorsal and ventral atrioventricular cushions. It results in a condition in which the common atrioventricular canal is never partitioned into the right and left atrioventricular canals so that a large hole can be found in the center of the heart. Ebstein's anomaly is caused by the failure of the posterior and septal leaflets of the tricuspid valve to attach normally to the annulus fibrosus. Instead, they are displaced inferiorly into the right ventricle. Foramen primum defect is caused by a failure of the atrioventricular septum to fuse with the septum primum. It results in a condition in which the foramen primum is never closed and is generally accompanied by an abnormal mitral valve. Tricuspid atresia is caused by an insufficient amount of atrioventricular cushion available for the formation of the tricuspid valve. For more details on these clinical conditions, check the video on congenital heart defects. Now, coming to the development of ventricles, the common sources contributing to the formation of the ventricles are interventricular septum and the atrioventricular canals. The source which contributes to the formation only to the right ventricle is the proximal 1/3 of bulbus cordis, and the source which contributes only to the formation of left ventricle is the primitive ventricle. Before we proceed to the formation of the ventricles, we will discuss on the septum formation in the truncus arteriosus and the conus cordis, which is necessary to understand the formation of interventricular septum. Neural crest cells originating in the edges of the neural folds in the hindbrain region migrate through pharyngeal arches 3, 4 and 6 to the outflow region of the heart tube. So during the 5th week, The neural crest cells form a pair of ridges arising opposite to each other, called bulbar ridges or cushions or swellings in the conus cordis and the swellings in the truncus arteriosus. Together, they are called conotruncal ridges. Since the truncus arteriosus is bent and not vertical, the truncus swellings or cushions arise on the right superior wall called the right superior truncus swelling and on the left inferior wall called the left inferior truncus swelling. When the truncus swellings appear, similar swellings or cushions develop along the right dorsal and the left ventral walls of the conus cordis. At the junction of the two parts, the swellings appear ventrodorsally. The right superior truncus swelling grows and fuses with the conus swellings on the left ventral wall of the conus cordis. The left inferior truncal swellings grows and fuses with the conus swellings on the right dorsal wall of the conus cordis. They also approach one another and fuse in the midline to form the spiral aorticopulmonary septum, separating the aorta from the pulmonary trunk. Since the ridges are spirally oriented, the relative positions of the aorta and pulmonary trunk are also spirally arranged. After complete fusion of the ridges forming the aortopulmonary septum or conotruncal septum, it divides the truncus and conus into an aortic and a pulmonary channel. Caudally at the conus, the anterolateral portion, that is the pulmonary channel, forms the outflow tract of the right ventricle called infundibulum, and a posteromedial portion, that is aortic channel, forms outflow tract of the left ventricle called aortic vestibule. In the adult, because of spiral partition, the relationship between the aorta and pulmonary trunk varies caudocranially. In the caudal part, the pulmonary trunk is anterior and aorta is posterior. In the middle, the pulmonary trunk and aorta are placed side by side, and in the cranial part, the aorta is anterior and the pulmonary vessel is placed posteriorly. We have seen that the neural crest cells form the conotruncal septum. These cells also control cell production and lengthening of the outflow tract region by the secondary arc field. Therefore, to see the clinical aspects, outflow tract defects may occur by several mechanisms. Defects in the secondary heart field, defects in the neural crest cells that disrupts the formation of the conotruncal septum, defects in the neural crest cells that disrupt the signals to the secondary heart field which they regulate. Heart defects caused by these mechanisms include pulmonary stenosis, persistent or common truncus arteriosus, and the transposition of great vessels. Since neural crest cells also contribute to the craniofacial development, it is not uncommon to see facial and cardiac abnormalities associated in the same individual. Coming to the development of second group of valves, the semilunar valves of aorta and pulmonary trunk. When the partitioning of the truncus is almost complete, small tubercles, which are the primordia of the semilunar valves, become visible on the main truncus swellings. One of each pair is assigned to the pulmonary and aortic channels respectively. A third tubercle appears in both channels opposite the fused truncus swellings. Gradually, the tubercles become excavated or hollow out at their upper surface, forming the semilunar valves. Recent evidence shows that the neural crest cells contribute to the formation of these valves. Formation of the semilunar valves is complete by the end of the 9th week. The aorta has one posterior valve and two anterior valves, above which the right and left coronary arteries arise. The pulmonary trunk has one anterior and two posterior valves. We have come to the last part of partitioning of the heart, septum formation in the ventricles. By the end of the 4th week, the two primitive ventricles begin to expand. This is accomplished by continuous growth of the myocardium on the outside and continuous cavitation and trabecular formation on the inside. The medial walls of the expanding ventricles become opposed and gradually merge, forming the muscular interventricular septum, which project upwards from the floor of the ventricles. This septum is composed of thick muscular tissue. When this muscular septum is complete, a large opening still remains between the two ventricles. This space is present between the free rim of the muscular interventricular septum and the fused endocardial or atrioventricular cushions as seen in the sagittal and coronal sections. This forms the interventricular foramen and permits communication between the two ventricles, and also it communicates with the atria through the atrioventricular canal. Meanwhile, the spiral subendocardial thickenings, the bulbar ridges, appear in the distal part of the bulbus cordis. The bulbar ridges then grow and fuse to form a spiral conotruncal or aortopulmonary septum. With the formation of the aortopulmonary septum, the size of the interventricular foramen becomes smaller. During further development, the septum intermedium formed by the fusion of the endocardial cushions proliferates or outgrows the tissue along the upper margin of the muscular interventricular septum, and this closes the interventricular foramen. This proliferating septum intermedium also fuses with the abutting parts of the aortopulmonary septum. Complete closure of the interventricular foramen forms the membranous part of the interventricular septum. So, the membranous part of interventricular septum is formed by the proliferation of the endocardial cushion along with the caudal part of the spiral or conotruncal or aortopulmonary septum. The closure of the interventricular foramen not only shuts off the path of communication between the right and left ventricles, but also ensures that the right ventricular cavity communicates with the pulmonary trunk and the left ventricular cavity communicates with the aorta. Fusion of the outflow tract cushions results in separation of the blood flow. The blood exits from the left ventricle through the aorta and from the right ventricle through the pulmonary artery. In addition, the right atrioventricular opening now connects exclusively with the right ventricular cavity and the left atrioventricular opening with the left ventricular cavity. This animation shows after the formation of aorta and pulmonary trunk, the blood flows from the right ventricle to the pulmonary trunk and from the left ventricle to the aorta. Now coming to the overall development of the interventricular septum, the interventricular foramen is usually completely closed by week 7. The sources contributing to the formation of the interventricular septum are muscular part from the muscular interventricular septum, membranous part from the proliferation of endocardial cushions, and caudal extension of conobulbar septum. We have seen the development of partitioning of the primitive heart tube until now, and what they develop into and how they develop. Now coming to the clinical aspects of the development of the arterial end of the heart tube. Ventricular septal defects are the most common type of congenital heart defects, accounting for approximately 25% of cases. Ventricular septal defects occur more frequently in males than females. Mostly seen ventricular septal defects are muscular ventricular septal defect, common ventricle, and membranous ventricular septal defects. Most ventricular septal defects involve membranous part, and many small VSDs close spontaneously during first year of life. Muscular ventricular septal defect is a less common type of defect that may appear anywhere in the muscular part of the septum. It is caused by single or multiple perforations in the muscular interventricular septum. Common ventricle, called cor triloculaire ventriculaire, is caused by failure in the formation of both membranous and muscular interventricular septum. Membranous ventricular septal defect is caused by faulty fusion of the right bulbar ridge, left bulbar ridge, and atrioventricular cushions. It results in a condition in which an opening between the right and left ventricles allows free flow of blood. With this, we have completed the partitioning of the primitive heart tube. Now we will discuss the formation of the conducting system of the heart and the fibrous skeleton of the heart. Formation of the conducting system of heart. Contraction of the heart by myogenic activity begins at about 28 days. The conducting system of the heart, which includes SA node, AV node, bundle of His, and Purkinje fibers, consists of specialized cardiac muscle cells. The SA node is derived from the neural crest cells, and the rest of the conducting system is thought to be derived from cardiogenic mesoderm. At week 5, initially, the pacemaker of the heart developing into SA node lies in the caudal part of the left cardiac tube or the heart tube. Later, the sinus venosus assumes this function. Cardiac myocytes in the sinus venosus region of the primitive heart tube begin to undergo spontaneous electrical depolarizations at a faster rate than cardiac myocytes in other regions. As looping of the heart occurs, the sinus venosus is incorporated into the right atrium. The pacemaker tissue migrates and lies near the opening of the superior vena cava, and these fast rate depolarizing cardiac myocytes become the sinoatrial node. The atrioventricular node and bundle of His are derived from two sources: cells in the left wall of the sinus venosus and cells from the atrioventricular canal. Once the sinus venosus is incorporated into the right atrium, these cells lie in their final position at the base of the interatrial septum. Now coming to the formation of the fibrous skeleton of the human heart, it is composed of several parts which are formed from different extracardiac sources. The components of the fibrous skeleton are 4 fibrous rings, aortic, pulmonary and 2 atrioventricular rings. Between these rings, there are interconnecting areas, the right and left fibrous trigones, central fibrous body and tendon of conus. The fibrous rings are formed by invagination of sulcus tissue at the atrioventricular and bulboventricular transitions. The central fibrous body, which is the interconnecting area, is formed from an ingrowth of tissue from the dorsal mesocardium and sulcus tissue. During valve development, the atrioventricular endocardial cushions, initially situated between the developing atrial and ventricular parts of the central fibrous body, are almost completely removed downwards into the ventricular cavity. A small portion of these cushions remaining on top of the ventricular septum is trapped by the surrounding sulcus tissues and becomes incorporated in the central fibrous body. These embryological findings on this centrally located area of the heart have implications for certain types of cardiac abnormalities. So with this, we have completed the development of the heart. Thank you for watching till the end. If you find the video useful, like, share and subscribe to the channel.